So now let's go ahead and start talking about nucleic acid hybridization. We're going to start by looking at the components. We're going to go through the steps of the process, and then I'm going to draw it out at the end. So the first component that you need are some cells, and we call those our reference and our experimental sample. Your reference sample would be healthy cells, as an example, and your experimental sample would be cancer cells. So that's one way that we could use it to compare healthy cells and cancer cells, reference, experimental. And we are going to use mRNA from these samples, which is going to be converted to cDNA, which is complementary DNA, complementary to that mRNA. We're also going to have a microarray, which is some solid surface that has a bunch of DNA probes on it. And a DNA probe has a single-stranded DNA sequence on it that's unique. And you'll have a couple of each type, and we'll talk more about that in a second. You're also going to have fluorescent labels for your DNA, cDNA for every single cell type. That way you could tell them apart. All right, so we're going to just go through the steps really quick right here, and then we're going to go through each one of them individually. So first, we want to make our microarray. Then we're going to select our cancer and our healthy cells. We're going to take the mRNA out. We're going to change it to cDNA. We're going to label the cDNA, mix the cDNA, put it on a microarray, and then scan the microarray. So we're going to start at step 1 and go through each of these and then draw out the process. Step 1, make a microarray. So remember, a microarray is some solid surface that has a bunch of DNA probes on it. And your DNA probes is a sequence of DNA that's single-stranded. So if you have a bunch of these single-stranded DNA probes, something else that you put in there that would also be single-stranded could bind. And whenever it's bound, you could tell, okay, this sequence is complementary to this sequence, so we know that this is in our genome if it's bound there. And we're going to have a couple of each type of DNA probe. That way you could have some of the cancer cells cDNA bind and some of the healthy cells cDNA bind. We wouldn't want to only have one because then only one of those would be able to bind. All right. Step 2 is selecting your cancer and your healthy cells. So our cancer cells would be our experimental sample, and our healthy cells would be our reference sample. So we would take both of those and we would have them. And then we would want to take out the mRNA. So you'd remove the mRNA from the cancer cells and the healthy cells. And you might be asking, because in the next step, we're going to convert it to cDNA, why even have mRNA? Why not just take the DNA out? And that's because whenever we're looking at cancer or healthy cells, we want to see what they're actually doing and what they're expressing. Is there a different protein that the cancer cells are expressing that the healthy cells aren't? Or is there a protein that healthy cells are expressing that cancer cells aren't? And the only way to do that is to actually look at the expression of the genome. And we know that mRNA shows us that expression because it's showing what's being transcribed, what's going to be translated into a protein. If we were to just grab a bunch of DNA out, you might be looking at this huge portion of it that's never even going to be expressed. So that's why we use mRNA, because we know it's being expressed. And then we're going to change into cDNA. So cDNA, as a reminder, is complementary DNA. And the way that we'll do this is with reverse transcriptase, which is an enzyme, and DNA nucleotides. And you might be wondering, okay, we have mRNA, why are we changing it back to cDNA? What's the point? And that's because DNA is a lot more stable than RNA, and so it's easier to work with because it doesn't have that 2' OH. So we want cDNA just because it's easier to work with. It's a lot more stable. So now we have cDNA, and now we want to label that cDNA so we can tell which one's from cancer, which one's from healthy cells. And the way that we're going to do this is with fluorescent bases, so fluorescent A, T, G, or Cs in your cDNA. And you're going to use one color for the cancer cells and another color for the healthy cells. That way, at the very end, when you're scanning it, if you see red, you can say, oh, that's cancer. And if you see green, you could say, oh, that's a healthy cell. And for example, red is cancer, green is healthy, but really you can choose whichever you want. All right, so now we have our cDNA labeled, so we're going to mix it all together. So we're going to take our cancer and our healthy cell cDNA, put it in the same vial. And remember that this isn't a big deal because we can tell them apart. If we were to scan, we'd be able to see which ones of those are cancer and which one of those are healthy. And that's why we're allowed to mix after we labeled them. So now that we've mixed them, we're going to put them on our microarray that has all of those little DNA probes. And anytime that cDNA is complementary to the microarray, it's going to bind. And then after that cDNA has had a chance to bind, we're going to scan the microarray looking for the different colors. Because scanning that microarray is going to pick up the signal from the fluorescent basis and give you some color. So going back to our previous example, we said cancer was red and healthy cells were green. We're going to keep working with that. And if you were to see some DNA probe that it's only red, you could say, okay, this gene or this sequence is only being expressed in cancer cells. If it was only green at that DNA probe, you could say it's only being expressed in healthy cells. And if it was yellow, you could say, oh, it's expressed in both healthy and cancerous cells. So the reason why this is important is because you could look at that microarray and see which sequences are expressed more in cancer cells. Once you figure out those sequences, now you kind of know their function. They're doing something to possibly cause the cancer. And then you can go back, maybe mutate that sequence and keep doing some other analyses. Whereas in your healthy cells, if you have something that's only being expressed there, you might think, okay, how can we get that sequence into a cancer cell? Would that fix it? So these are really important, seeing the expression of the genome in cancer versus healthy cells. Remember, that expression is talking about your mRNA. So now that we've gone through those steps, let's draw this process. And we are going to start with a microarray that has a bunch of different DNA probes on it. So each of these lines represents a DNA probe that has some single-stranded sequence of DNA on it. Remember, they're going to have multiple of each type, because if both the cDNA from the cancer and the healthy cells has a sequence that can bind, we want them both to be able to bind. That way we can see, okay, they're both being expressed. So we've made our microarray. Now we're going to select our cancer and our healthy cells. So I say that that's my cancer cell that I selected, and here is my healthy cell. And I'm going to take mRNA out of both of these. And remember, the reason that we're using mRNA is because we want to see what is being expressed in cancer versus healthy cells. And the way to see what's being expressed is using the mRNA, because that's what's being transcribed. That's what will turn into proteins, and the proteins are going to be doing things in the cell. Unfortunately, though, our RNA is not very stable, so we want to change it to cDNA for both of them. And that cDNA is just going to be complementary to the RNA. We'll use reverse transcriptase, we'll use DNA nucleotides, and now we have our cDNA. And within that cDNA, we're going to have fluorescently tagged bases to where it would appear red for a cancer, as an example, and green for our healthy cells. So now that we have everything labeled, we can mix it all together. So I'm just going to draw them in a big vial together. We have them mixed together, and we're going to take that vial, and we're just going to pour all the cDNA on our microarray, spread it all out. That way, our cDNA can bind, because our cDNA is going to be single-stranded at this point, and so it can bind to the other single strand if they're complementary. And then if it stays there, it'll be stuck through the hydrogen bonding. And whenever we scan it afterwards, we can see, oh, is it tagged red? That means it's expressed in cancer cells. Is it green? That means it's expressed in healthy cells. Or is it yellow? That means it's expressed in both. So let's just do a visual of that. Let's say that our cancer cells bound to these, and our healthy cells bound to these. Or I should say our cancer cDNA bound to those, and the healthy cDNA bound to these. And we'll say we have a mix as well with yellow at some of these. And remember that those DNA probes, the sequence is known. So I could go and look at one of these probes and say, let's say only one of them was expressed in cancer cells, and that was the only difference, and the rest were yellow. So the only difference in sequence expression was that one cancer cDNA. I could go and see what probe that's bound to, and then knowing the probe sequence, know what the cDNA was, know what the mRNA was, know what the DNA sequence in the cancer was that was being overexpressed. So any of these, you know the sequence, meaning that you can go back to the cell and say, okay, this is the sequence that I know is causing overexpression or underexpression or whatever you need to do there. So nucleic acid hybridization is a really important first step in looking at some of the differences between cancer cells and healthy cells or whatever else you want to study. So I'm going to go back to the step slide really quick. We'll run through it one more time. So we made a microarray, which has the DNA probes on it that are going to be single-stranded and complementary to whatever you're looking at. We selected our cancer and our healthy cells, and we took the mRNA out because we're looking at expression, but mRNA is unstable, so we changed it to cDNA. We labeled the cDNA with fluorescent bases. We mixed them together. We put it on our microarray, and then we scanned it to see where each one was being overexpressed, underexpressed, or equally expressed. All right. And that is the end of nucleic acid hybridization.
I hope you found this video really helpful. The concepts and information presented in these videos will be true no matter what genetics class you are taking. However, the concepts presented in this video are referencing material currently covered in Baylor University's coursework. Remember, if you are currently enrolled as a Baylor student, we offer free tutoring services. Our tutoring center is located on the first floor of the Sid Richardson building. You will find all the details you need to know about these services on our website, www.baylor.edu/tutoring. You may schedule a free 30-minute one-on-one tutoring session online through Navigate, or just drop in during our open business hours. For more information about our current services, please visit our website. Thank you.